Alright guys, welcome to one of our middle lessons on DNA and genetics. I'm going to try and rush through a bunch of videos tonight, so that way you guys can get all the printed notes for your test, which will be on Friday. So, let's do this. Things are going to be a little bare bones. Sorry, probably won't be as many memes after this one. All right, so basically when Gregor Mendel was doing all of his research on pea plants, he noticed certain patterns. He used those to develop what we now call Mendel's laws. And they are very reliable laws that help us understand how genes are inherited. So genes are particulate, meaning that they're carried in they're carried in a certain location, so they're not just kind of like, oh, you get this gene by having these 550 segments of a chromosome. They're like a single particle. All right, so your review on genotype versus phenotype. Phenotype is your physical appearance. Now, usually what happens is genes are going to determine the phenotype by making a particular protein. That protein has a function and it affects the appearance or the working of the organism's body. However, if you're looking at a dominant versus a recessive gene or allele, then what usually happens is the recessive allele is a mutant that no longer makes a functional protein, or it can affect where it's expressed or something like that. So usually a recessive is we're not making the protein. The dominant trait is we are making the protein. This explains why heterozygotes tend to have the recessive trait covered up. Because usually you can get by making only half the protein you need. That's enough to help you develop whatever trait you're trying to develop. So there is an actual genetic basis for a lot of the traits that Gregor Mendel was studying. For example, one of the things he studied was wrinkly peas. So those wrinkly peas on the right actually have a mutation in a specific enzyme, and we've actually tracked that mutation down. It's due to the addition of an extra 800 nucleotides that were added in there, so it's probably some sort of insertion mutation, and that basically messed up an enzyme that makes starches. Starches aren't made correctly, and so then you get wrinkly peas. So alleles are only going to be created by mutations. So remember, mutations are entirely by accident. Mutations are not desirable circumstance, because most mutations end up being negative mutations or harmful mutations. Some mutations are neutral, and a very few are beneficial. When we're talking about the allele that was probably the original allele before a mutation caused a new allele, and is present in most of the population, we call that the wild type allele. So for example, what I said in class is our wild type allele for eye color or hair color in humans is most likely to be brown or black for hair, because that's what most of the population has, and it looks like all the other hair and eye colors arose from mutations of the original brown. If you have more than one allele that a particular gene can have, you say that gene is polymorphic. So when we're doing these genetics problems and all this stuff with genetics, we're all working with only polymorphic genes. Because if a gene only has one allele, well, you don't need to do all the fancy Punnett squares and probabilities to figure out what the baby's going to inherit. They're going to just get that one allele that exists. So let's talk about how we label our generations. So you may have seen some P's and some F's as you were going through these genetics problems. This all comes kind of from Mendel's work, where he was looking at ways to refer to specific generations. So he would usually start out with two purebred strains, meaning he would say take peas that had purple flowers and mate those with peas that had purple flowers, and then take those offspring and mate them with peas that had purple flowers, until he was pretty sure he had a true breeding strain. He would do the same thing for white flowers, although since that turned out to be a recessive trait, it's pretty easy to isolate. So once he had these true breeding strains, he called them the P generation or parental generation. Now that we understand the kind of biochemical nature of genetic inheritance, we know that his true breeding strains were homozygous for whatever trait he was looking at. And then he took these two pea plants and he would make them breed. And so the resulting offspring were called the first filial generation, filial literally meaning son. And these guys all ended up, we know now, and all ended up being heterozygotes. So they would look identical and they would all have the same genotype. Then what he would usually do is he would take those F1 plants from the first filial generation, he'd mate them with each other, and he would produce a second filial generation. And that's where we get that kind of famous 3-to-1 ratio for dominant versus recessive phenotype. And he was producing, especially in our F1 generation, he was producing hybrids. A hybrid is just, in general, a hybrid is a combination of two or more things. In genetics, a hybrid is basically you take two organisms with different traits, you mate them, and the offspring are hybrids. So the simplest type of cross is just your regular old monohybrid cross. I love this diagram because it does make sure that you remember, hey, you've got two alleles from each parent. Make sure to show you the male and the female signals. And it also is showing you that in the gametes, you can divide up, these alleles, so they're going to go into different gametes. So for example, up here, the homozygous dominant P plant is going to be giving 2 copies. It's going to be giving 2 copies of the dominant allele. But so that's all it can give to its sperm in this case. The wrinkly pea plant, the only thing it has to give is recessive alleles. And then we form our Punnett square. And then just note that from here, you're actually going up to here. Because this is the P generation. This is your F1 where you're basically taking those, well, this is the F1 generation, they're the heterozygotes, and then your F2 generation, you're crossing those heterozygotes. So from his work, when he was looking at this, he started to develop this idea that the copies of a gene that you have, the 2 copies that we have, are going to separate from each other and during the process of creating these gametes. So he back then didn't know the genetic basis, the chemical basis, like what was going on. He just knew there was some factor that gave traits, and depending on, so what he was basically saying is some people were like, oh, hey, you take purple flowers and white flowers and you mate them and you get purple flowers, that white trait disappeared. But by doing the second filial generation, he proved that that trait didn't disappear. So it must have still been passed down in some form. So he came to the conclusion that, well, however many copies of a trait you have, one goes into one gamete, another one goes into another gamete. So if you are heterozygous, for example, half of your gametes will be dominant, half of your gametes will be recessive. And it's really quite amazing that he came to all these conclusions, considering back then they didn't even think that DNA was the genetic material. So he had no clue what the genetic material was. And he was still figuring out all these things that hold true with what we've discovered by studying DNA. So Punnett squares are useful because they help you consider all possible combinations. They can be your best friend, but they can also be your enemy. I'll show you how later. And just in case you've forgotten, for a Punnett square, you're basically bringing down the letters. So I'm bringing down that recessive R. Then I'm going to bring across my letters. And I'm going to bring across my letters. Now, I want to point something out. This guy is the same as this guy. The only thing that's different about them is the order in which I put the alleles. Technically, it doesn't affect the genotype or the phenotype of the individual you're looking at. It doesn't affect them, the order in which you put those dominant and recessive alleles, but by convention, in genetics, what we always do is we always write the capital first. So that's why you have 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive from this cross. If you don't know the genetics, the genotype of an individual, you can always do a test cross. I explained in class why this is not always feasible though, because you need something with a fast enough generation time that you can get results, and you also ideally need something that produces a lot of offspring. So let's say we have this purple flower, and we know purple trait is dominant, but we don't know if it's purebred or if it's a hybrid. We don't know if it's homozygous or heterozygous, and there are cases where this could be necessary to figure out. So what they would do is they do a test cross. They would breed it with a recessive plant, because if you think about it, a recessive plant, homozygous recessive and homozygous dominant, all of their offspring are going to have the dominant phenotype because they're all going to be heterozygous. Whereas if this guy right here was heterozygous, half of his offspring should have the recessive phenotype, and you should be able to see that. So pretty ingenious way to figure it out. Now, let's talk dihybrid cross. In a dihybrid cross, you are looking at more than one trait. You're looking at 2 traits now. So here in this example, we're looking at the color of the peas, and we're looking at whether it's wrinkled or not. And I want you to pay attention to this process up here. It's showing you the different combinations of traits that you can have in a particular gamete. So let's go over why that is the case. So we have our genotype, which is heterozygous for everything, right? Now, in a gamete, each of these traits is going to separate and go into different gametes. However, we know that every gamete should end up with one of the alleles for R for that trait and one of the alleles for Y. And if you're wondering how you figure that out, well, the way you figure out which gametes are kind of mixed together is FOIL, which I hope you learned in math class. It stands for first, outer, inner, last. And it's a way of combining these. So when I say first, I'm going to take my first 2 letters and I'm going to put them together. So R, big Y. Outer, I'm going to take my big R and I'm combining it with my last letter, which is a little y. Then I'm going to do my inner, which is that little r, and I'm going to combine it with my big Y. And then finally, I'm going to do my last, which is going to be my little r, little y. I want to point something else out. I know that I did not put these capital first, but that's because they're 2 different genes. If they're 2 different genes, you write the alleles for them in alphabetical order. So notice in all of these, I've alphabetized my alleles, and then I've written them in individually within each gene. I've written like the uppercase ones first. So notice right here, you've got big R, big Y, little y. It's not big R, little y, big Y. Here, you've got everything set up alphabetically, so your R's are clustered together, your Y's are clustered together. Now, what we did up here is we showed the 4 possible gametes this plant can make. So, it can give you an allele for yellow and smooth. It can give you the alleles for yellow and wrinkled. It can give you the alleles for green and smooth or green and wrinkled. So keep in mind, this is the combination you can have. You can have both traits dominant, both traits recessive. The first trait could be recessive and the second dominant, or vice versa. So how do we figure out all these probabilities? Well, you could, if you have plenty of time and nothing better to do a giant Punnett square. However, it is like really time consuming and you don't have time for that on the AP test. So here's another method you can do. Giant Punnett square. So let's say your cross was 2 heterozygotes. And you wanted to know the probability of them also having a double heterozygote. All right, let's make it simple. Let's say the probability of them having a double recessive, homozygous recessive all the way. Okay, so what we do then is let's look at this first pairing. What is the probability, or sorry, I'm doing probability already. So what we can do is we can look at this first pairing of the A alleles. And then we can make a Punnett square for that, which is down here. What's the probability that we're going to get a homozygous recessive baby? Well, we got 1 out of 4, so that's our probability. Now, if we look at our second gene for our B alleles, what's our probability of having a homozygous recessive baby? Again, it's 1/4. And the probability of having both of those is going to be those numbers multiplied. So it's 1/16. I highly recommend you stay in fractions, because if you start doing this in decimals, it's much harder to convert from decimals to fractions than it is from fractions to decimals. It takes more work. So leave it in fractions. If you are looking at an answer choice and everything's in decimals, then you can just divide it out on your calculator. All right, so quick recap of probability. So an event that is absolutely certain to happen has a probability of 1. So unless like weird catastrophic stuff happens, the probability of the sun going down in the evening and rising in the morning is 1. Events that can't possibly happen have a probability of 0. So for example, getting more energy out of a system than you put in, or like producing energy out of nothing, we'll say that. So talking about violating a law of conservation of energy. That's impossible, so that has a probability of 0. And anything else, depending on how likely it is to happen, has a probability between 0 and 1. Now, just keep this in mind. This is what you're going to use most of the time, but not always. The probability of 2 independent outcomes, 2 independent events happening together, is found by multiplying their individual probabilities. And that's what we've already been doing. All right, so straight up probability methods. We can use this to do any type of cross, all the way up to a tetrahybrid cross. So we did this in class, but let's do it again. So I have 2 individuals. They are heterozygous for 4 different traits. And I want to find out what is the probability that they're going to be all recessive. So what you can say to yourself is, what is the probability of this parent giving the A allele? Well, that probability, and I'm going to highlight all the alleles that we're looking at the probabilities for, just so I don't have to keep going back and forth between the pen and the highlighter, because it's a pain. All right, the probability of getting that recessive allele is 1/2. It's 1 out of 2 chance. Out of this capital B and lowercase b, the probability of my getting the recessive allele is 1/2. So I got a 1 in 2 chance. Same thing for the next couple of alleles. Same thing for the next couple of alleles. Okay? And then same thing for the next guy, 1 in 2 chance. Same thing for the next guy, 1 in 2 chance. And you see our pattern. To find out the probability of all of these happening together, I got to multiply them together. Now, to multiply a bunch of fractions, you basically take your fraction line and you extend it across the whole thing. So what's 1 times, and then you multiply across, and you do that on the top and the bottom. So what's 1 times itself? No matter how many times you multiply 1 by 1, you could do 1 by 1 by 1 by 1, or multiply 1 by 1 like 100 times, it's still going to always equal 1. Too bad it's not the same for the number underneath, because that's 1, 2, 3, 4, 5, 6, 7, 8 2's being multiplied, which I believe we said was 1/256. So not very likely. So we talked about a test cross. We can't always do a test cross because humans don't like to be bred with each other. And And also we have very slow generation time, so we'd be terrible for those experiments. However, we can look at inheritance patterns by looking at the different offspring being produced within a different family. We can even trace diseases, and it's especially important if we know that whether it's a dominant trait or a recessive trait. If it's a dominant trait, then everybody who's got that phenotype has to have at least one parent who has that. Let's see. And then if it's recessive, then you can often have like two parents who don't have that phenotype. They don't have that trait, but their children have it because their parents are carriers. They're carrying the allele, they're not showing it off. So here you have an example of dominant inheritance. Notice everybody who has it got it from a parent. Their parent has to have it. Notice this chick right here doesn't have it. Let's say she married somebody who also didn't have it. None of her children would have it. So she would have all unaffected children because she doesn't carry the trait, so she can't pass it on. All right, recessive patterns though, on these types of pedigrees, what you'll see is these shaded in circles or squares, and that is going to be your carriers. By the way, the circles are females and the squares are males. And if you're wondering, ew, why is there a mating between relatives on here? It's because it's showing a royal family, and that stuff happened in the past. Anyway, you'll see that out of this entire family, you only have one affected male. And if you're curious, this was actually the royal family of, including Russia, and this was like young Prince Alexander or Alexei, and then this was Anastasia, who had that movie, at least one movie made about her. But anyway, so they're tracking a recessive trait going through that family. So, very quickly, you might ask if you have something that is basically like a recessive disease, so let's say sickle cell, which is a recessive trait, why would you keep that trait around in the population? If it's a disadvantage to have that disease, why would it stay around? Turns out there's a reason for that. It has to do with evolution. Turns out that being a heterozygote for that gene gives you a chance, extra chance of survival in some other way. So carriers of the sickle cell gene are actually fitter in tropical regions like Africa or India because there are a lot of mosquitoes there who can carry malaria, and those heterozygous individuals aren't as easy to infect. So malaria parasite can't infect them as easily, and they are able to survive and not die of malaria. On the other hand, if you are homozygous dominant, so let's say you're homozygous dominant, oh, well, you don't have any sickle cell going on, but you're going to get killed by malaria. You're homozygous recessive, yeah, you just died without medical treatment. And then if you're heterozygous, you are just healthy enough with the sickle cell problem, because some of your cells will be sickle shaped perhaps, but it's not enough to cause like an actual health problem. I believe most people who are carriers of sickle cell don't even end up having to go to like the doctor very often for blood transfusions like people who actually are affected by sickle cell anemia. But they are more resistant to malaria, so they have a survival advantage, and that keeps that recessive around in the population. Also goes to show some crazy genes have crazy advantages here and there. Sickle cell, anybody who has it would tell you it's not a pleasant disease to have. It is going to affect you your whole life. And you're like, why? Why would this terrible gene be kept around? Turns out it had an extra advantage in some other, like, seemingly unrelated event. And so you never know what alleles, what variation will be beneficial in a different environment. It's just crazy. All right, so that is called heterozygote advantage, where being a heterozygote gives you an advantage over either of the homozygotes, and so that allele sticks around. Tay-Sachs disease is something I am supposed to teach you about based on the lesson planning guide. don't remember what its significance was here, but basically it's recessive, it's on an autosome, and it is, I believe, let's see. I mean, it's obviously maintained in the population. Well, let's skip it and move on. We did this problem in class, but I'll do it again just so you can watch it multiple times. A man has a dimple and brown eyes. By the way, we've got some information down here, and I like to start writing notes while we go. So dimple is dominant, brown eyes are dominant. Okay. So, man's got dimples. We know he's got to have one dominant in there, and brown eyes. We know he's got to have one dominant in there. So by the way, D, that's for dimples, and B is for eye color. Okay, so we do know though that his father had blue eyes and no dimples. Well, the significance of his father having blue eyes is he had to have a recessive, so he had to give that recessive to his son. Same thing with the dimples. And then he marries a woman who has same situation. She's got a dimple, she's got brown eyes. If we go back and look at her family history, her father had blue eyes, so he had to give her a recessive. Her father had no dimples, so he had to give her a recessive. We want to know what the probability is that their first child will be blue-eyed without a dimple. So we want to know the probability of a totally recessive baby. I'm going to do this probability-wise just because I think that's what y'all need the most practice on. The probability of them getting the recessive allele from their dad for dimples is 1/2. For their dad, for the eye color, it's 1/2. From their mom for the dimples, it's 1/2. And from their mom for the eye color, it's 1/2. So to find out the probability of them getting all of those gametes, you got to multiply across, and remember, 1 times 1 times whatever is always 1. And then at the bottom you have 2 times 2 times 2 times 2. Stick it in a calculator, you get 16. So they have a 1 in 16 chance of having a double homozygous recessive baby. All right, you have multiple types of dominance, so it's not always one trait completely covers up another trait. You can also have incomplete dominance. The cross you see here has an incomplete dominance for flower color. So notice if you breed red and white flowers of this variety, they're actually going to make pink flowers because the dominant does not completely cover up the recessive. So here you have 3 phenotypes. You've got a dominant phenotype, a recessive phenotype, and you've got that hybrid phenotype. Codominance is when you have a couple of alleles, they cover up another allele, so there's going to be a recessive, and then when you have those codominants, they both show up in the phenotype. So you're going to be, able to see them both. Sorry, cat problems. Okay, so this is another problem that we did in class, but I'm going to do it here so you guys have it to look at. So A and B blood type are dominant over O. A and B are codominant with each other. So we've got an explanation of which blood type has which phenotype, etc. We've got a man who's type A, marries a woman with type A. So we know he's got to at least have one A in there. And she has type A as well. Their first child gender unknown, has type O blood. How could that be possible? Well, child had to have inherited an O from each parent. So, each of the children, the child has type O blood, parents each have AO. All right, let's look at this pedigree. It's a little different. Notice that you can have a normal male, a normal female, an affected male, and a carrier female. What you don't see on here is there's no carrier males, and you don't really see any affected females. This type of trait, if you see this strange pattern, where it shows up more in one gender over another, so notice, no female is affected in this family, but 2 of the males are affected. If you see that kind of trend happening over and over, what you have is a sex-linked trait. Sex-linked traits are generally carried on the X chromosome. Notice the X chromosome is much larger than the Y chromosome, so there are a ton of genes that are found on the X and not on the Y. They don't have a homologue there, so you only actually, if you're male, inherit one copy of that gene. That means that they don't have as many chances to have a dominant gene to cover up any sort of recessive. So that's why there's some traits that get passed on more to men. So color blindness is a big example. Hemophilia is another example. So let's do a cross for this. So we have a white-eyed female fruit fly, and white eyes are X-linked and recessive. So she's going to be X little r, X little r. And we're going to mate her with a red-eyed male. So he's going to be X big R, Y. Because unless you're told otherwise, we're assuming that males have the Y chromosome, females have only X's. Okay, so if we do this cross, what percent of the female offspring will have white eyes? Okie dokie. Well, the only thing that the mom can give is the allele for white eyes. So we've got a 1 in 1 chance of that. How many recessive alleles can the dad give? Well, hopefully you answered 0. Multiply those two probabilities together, your probability is 0. This lady with that man, or this lady fly with that man fly cannot produce white-eyed females. An interesting thing about X chromosomes is the fact that even though ladies, we all have 2 X chromosomes in every single cell, to kind of get around the fact that we have that extra bits of DNA that a male doesn't have, we actually inactivate one of our X chromosomes in every single one of our cells. So what will happen is sometime during development, your cells will randomly inactivate one of the chromosomes, and it produces something called a Barr body. It's super condensed. It's probably only ever unpacked in time for DNA replication, and then it's packed back up. And so these cells that have a random Barr body inactivated will then go on to produce more cells by mitosis, and in each daughter cell, the same X chromosome is going to be inactivated. What that can end up showing you is, for example, in cats, and this picture happens to be one of my dearly departed cats. She was a calico, and a calico cat has different patches of color. So you'll notice that her nose looks very orangey, and she had those patches of kind of orange tabby fur. And then you'll notice it's a little harder to see here, but towards the back she's got more black and gray, and she had patches of more black and gray tabby fur. So what happens is one of her X chromosomes carried a trait for orange fur, and one of them carried a trait for black fur, and depending on which ones were inactivated, those would be expressed in different patches of her fur, and that was what results in the different color. It's pretty cool. It's the reason why most, if not all, calico cats are female. We also have some inheritance that happens outside of the nucleus. So remember, mitochondria also have their own DNA, and you inherit that independent of the nucleus. Turns out, vast majority of that comes from your mom. There's a little bit of evidence starting to come out that maybe a little bit of it comes from your dad, but since a sperm cell is very, very light on material, it's mostly genetic material, some mitochondria, and a tail, and the mitochondria are just there to power it long enough to fertilize an egg cell. Whereas an egg cell is very large, has all these materials for developing a zygote, and so it has tons of mitochondria, and that's where we get our mitochondria from our mom. So you could inherit, if there's a trait that's caused by some sort of mutation in a mitochondrial gene, that's something that you can inherit from your mother. You're not likely to inherit it from your father. Though I will say this, these traits that mitochondrial genes code for are pretty necessary to us performing cell respiration, so they're not likely to have a mutation in them. Chloroplasts are also inherited maternally, so if you have a certain plant producing an egg cell, it's fertilized by some different pollen from another plant, then any traits in the chloroplast will come from the egg cell. And then last but not least, some types of genes, the inheritance patterns are really complex. You have more than one gene affecting what the trait will be. So skin color is a very good example. There's multiple genes that are involved in skin color. I think it's actually more than the 3 that you see up here. And depending on the combination of them you have, you'll have different skin tones. Because skin color is in part due to the production of 2 or 3 different types of melanin. So that alone gives you 3 genes. And then there's a bunch of steps along the way of producing pigment that can affect how it's expressed and what skin color you are. So it gets pretty complex, which is why if you're wondering, well, we decoded the human genome, how come we don't know how everything works yet? Because it's complicated. All right, on to the next one.